In this class?
Don't we?
We haven't covered anything yet.
Isn't it like the week after next?
Yeah, it is. It's a Tuesday.
Okay, well, we'll see where we get to. As my eyes widen, I'm like, I've only covered one chapter. We're not ready for another test yet. Uh-oh. That's a problem.
We finished chapter 5.
We finished chapter 5 and chapter 6.
So that's like 1.5.
But chapter 5 was not planned to be included on the next test. So.
Yeah, I think it was 6, 7, 8, 9.
Yeah, we're not going to get through 7, 8, and 9. We might get through 7 and 8. We'll see what we can do. So that brings us to where we are today, which is starting chapter 7. And this is the other half of the T cell activation story, the major histocompatibility complex and antigen presentation. So when we start trying to activate adaptive immunity, B cells that make antibodies, that's part of the story. The other half of the story that's critically important are the T cells. And this is probably what I did the majority of my personal research on, were T cells and how the T cells got activated and what they did in response to viral infections in particular. For a T cell to be activated, it actually has to see its antigen, its little protein fragment, in the context of MHC. What that means is you have to have this other protein on the surface of your antigen-presenting cells. That's dendritic cells or macrophages. And that serves as a little pedestal that sort of holds your peptide, this orange-colored piece, and allows this to be detected by the T cell receptor. If you do not have this in complex with this other protein, the T cell can't really see it. It's going to ignore bits of protein that are just free floating in the environment. It's specifically looking for, it has to bind to this protein, and there has to be a little peptide fragment in there for it to actually recognize.
Do B cells need MHC as well?
B cells are independent of MHC, so they do not require this. This is unique to T cells. And that's important because one of the major subsets of your T cells are the cytotoxic or killer T cells. Their job is to kill infected target cells. They are one of the few types of cells in your body that literally has a license to murder you or parts of you, the infected cell parts of you. So you want this process to be carefully controlled and make sure that it's regulated and make sure that it's not happening when it's not supposed to happen, because otherwise you end up with major health problems. So we're going to start by looking at the structure and the function of MHC. There are two different classes, class 1 and class 2. This MHC stands for Major Histocompatibility Complex. That was on the title slide. In humans, this is also known as HLA, Human Leukocyte Antigen.
They're the same thing?
They are the same thing. Human leukocyte antigen.
So you're saying that this is also an MHC?
This is MHC, yeah.
It's just another name for it?
It's another name for it. In mice, you tend to see it called MHC. In humans, it tends to be called HLA.
Do you also call that HLA1?
Actually, there's different designations. There's HLA, DR, DP, DQ. No, it couldn't be that simple. There's a lot of different subtypes of this stuff. This is the basis for tissue typing. So if you think about organ transplantation, heart transplants, lung transplants, kidney transplants, liver transplants, any of the fairly commonly transplanted organs and tissues today, when they talk about having to have a tissue type match, they're talking about these molecules because they have to match what the immune system of the recipient has. If they don't match, two things are going to happen. One, the immune system's going to look at that and recognize it as being foreign and attack it. And that's the major concern because that leads to organ and tissue rejection and failure of the transplant. And number two, it also has an impact on the immune system itself because the immune system wouldn't be able to recognize antigens that are being presented on the surfaces of those transplanted tissues if they did get virus infected, for example. So it's also a potential route for infections.
Why is it called human leukocyte antigen?
Because you find these on white blood cells. Leukocyte. Leuko means white, cyte means cell. These were first discovered on white blood cells.
But why the antigen box?
Probably because it actually presents antigen.
It's like the foreign substance.
That is part of the foreign substance, yes. We're going to talk about how antigens get made and how they get processed. That's part of this chapter. Yeah. So these are members of the immunoglobulin superfamily, which means they have immunoglobulin domains. You're familiar with those. Those are those loop structures stabilized by disulfide bonds. There are two pieces to MHC1. And the first piece is the alpha subunit, and this has 3 domains on it, so 3 different big chunks that kind of make this L-shaped region on here. That larger protein chain is the alpha chain, so you have the alpha 1, alpha 2, and alpha 3 subunits. They're each about 90 amino acids long, so the whole protein is a little over 300 amino acids in length. There's a transmembrane segment that anchors this in the cell membrane. That's about 25 amino acids. And then you've got another subunit called, that's the one that's highlighted in red, that's called the beta-2 microglobulin protein. This associates with the alpha chains solely via non-covalent interactions, which we've seen a lot of also. So it just sort of hovers there through ionic and hydrogen bonds and van der Waals forces and all that stuff. What's important about the structure of this protein is that up here at the top, between the alpha 1 and alpha 2 subunits of MHC1, these 2 regions in particular form this sort of bed, I guess I would call it. So you've got this little spiral alpha helix on the edges of it, and you've got beta sheet that forms kind of a flat surface. So it creates this almost cup-like shape, and this is where your antigen is going to rest, is in this little slit right here. That's where your piece of your virus or your bacterium or your cell is going to go, is right in this groove that exists between the alpha domains of MHC1. The 2 alpha helices sort of form the walls, and the beta sheet section forms the floor of the cleft. You can fit in a peptide that's 8 to 10 amino acids in length in there, and there's a little bit of variation in that part of that. We'll talk about why that occurs. Longer peptides just simply don't fit, so that's why it's only 8 to 10 amino acids in size. Yes?
Your B2 microglobulin, is that, I know it's non-covalent, but is it permanent? Is it supposed to stick there, kind of like DNA sticks with its other chain?
It stays pretty well associated, so yes, it's a semi-permanent.
Why is it non-covalent?
Why is it non-covalent? Because it's a separate protein chain. It's encoded on a separate chromosome.
They don't have like disulfide bonds or anything, do they?
No, that would be covalent.
Which one was not?
Beta-2 microglobulin. The 3 alpha subunit portions, those are all part of one long continuous protein chain. So what's important about this? These MHC1 molecules, these interact with CD8 on T cells. So this is kind of like a ligand receptor interaction. MHC and CD8 and CD4 or CD8. For MHC1, it pairs with CD8.
Is it like there's the MHC molecules, like the long things that can attach to the antigen, or is that the CD8 part?
That is a co-receptor.
Yes, co-receptor.
Yeah. It took me a minute to figure out what you were.
I was trying to word it.
Yeah. No, you got there.
The alpha chains, and then the bottom part like the beta sheet in the picture.
If you think about what this looks like, it looks a lot like the T cell receptor already, with because in the T cell receptor, you had sort of the two variable chains and then the two constant regions. This is structured slightly differently, so you've got three alpha domains within this first protein and then a totally separate protein. Within these, you do see disulfide bonds. So they're within the individual domains, but not between alpha and beta.
Are they their own protein sheets?
Yes.
Like A3, A2, and A1?
Yes.
Oh, okay.
I mean, they're not each individual a separate protein. They're all part of one long continuous protein. It just wads up into three different distinct regions.
Oh, so it's like three binding domains or three domains?
Three domains, yes.
Oh, okay.
Yeah.
So one protein, three.
One protein, three domains.
Cool.
With a bunch of loops. Yeah.
Yeah.
MHC2 looks very similar, doesn't it? It is also a member of the immunoglobulin superfamily and therefore has immunoglobulin domains. It has an alpha chain and a beta chain as well. Both of these, in this case, pass through the plasma membrane. This is one of the things that was different. Beta-2 microglobulin was not anchored in the cell membrane. It was just free floating. If I go back for a second, I can show you that. However, since I'm actually recording today, I can't go back. It won't let me. That's nice. Okay. So the beta-2 microglobulin, if you go back when you look over this later, that one was not anchored in the cell membrane. It was just floating and associated with the alpha subunits. The other difference in this case is you have an alpha chain that is one protein, and it forms the two vertical domains on the right-hand side here. And then you've got a beta chain, which is also two domains that forms the left-hand side. So two discrete proteins, but overall, the structures are weirdly similar. They're put together a little bit differently, but the shape is the same. And that's because the function is basically about the same. Again, you get a peptide binding cleft or groove that actually forms between the walls of this and the flat beta sheet portion. This one, however, what's different about MHC2 is that it's actually open on the ends. So this actually creates a literal groove that isn't closed and boxed in. That's one of the differences between MHC1 and MHC2. MHC2 can actually accommodate longer peptides of 13 to 18 amino acids, as opposed to like 8 to 10. So this one can deal with longer segments of a protein.
And it's deal with longer segment because it's not closed.
Because it's not closed on the ends. Yeah, so those can just like drape over and hang over the edges, but that was not an option for class MHC class 1 because it was closed on the ends.
So for like massive proteins, like the spike protein and COVID is like what, like over 100 amino acids?
Right.
Will we just have like a T cell receptor that can take in like one chunk of that giant spike protein?
Yes.
Oh, is that how like even though they're totally random, you can still get something that will work with something?
Yes.
That makes a lot of sense.
Little like binding domains, parts.
Yes. So if you think about an overall protein, and this is something that we actually, that I wasn't really ready to talk about yet, but since it comes up, I will. If you think about a protein, it's supposed to be this sequence of like 100, 150, 300 amino acids long, some of them even longer than that. There's these big long chains of amino acids, right? But you're only going to fit 18 of them at a time in one of these, maybe 8, 10. Like that's a tiny little piece. And so what we're talking about when we talk about an antigen are those 9, let's call it 9 amino acids for MHC1, and I think optimum for MHC2 is somewhere around 13 amino acids for MHC2. You're only taking a tiny little piece of that like spike protein from COVID. That's all your MHC is going to bind to and recognize. But you're going to make a ton of variable MHC. Well, actually, your MHC itself is pretty constant, but it's able to accept a whole bunch of different peptides. So chances are, out of that whole, call it 100 amino acids long, that's like 10 different options for how you could recognize that protein if you took it in 10 amino acid chunks. That's not even counting like if you just did like, that's just 10 keeping it in one reading frame. If you did 1, 2, 3, 4, 5, 6, 7, 8, 9 versus 2, 3, 4, 5, 6, 7, 8, 9, 10, like there's a whole lot of possible combinations that you could potentially recognize.
Is that because it gets fragmented?
It does get fragmented. So the way that where those pieces of proteins, those peptides come from, is digestion and eating of the virus that occurs within the cell. We're going to talk about those pathways. So they're part of this chapter, where exactly the peptides come from.
Where's the, so the cleft is the binding site?
So the cleft is the binding site, yes. That groove is the binding site for the peptide.
And where is that?
That is right between the two alpha helices, so they form the walls of the channel, and the beta sheet arrow segment forms the floor of the channel.
And the same thing for
And it just creates a cup-shaped sort of groove.
same thing for MH1.
Same thing for MHC1.
MH2.
Yeah. MHC2 specifically is going to bind and interact with CD4 on the T cells. So CD8 is great, and you see that on cytotoxic T cells. CD4 you see on helper T cells. C equals cytotoxic. So you have two different classes of T cells in particular that we talk about the most often. This is defined based on whether they express CD4 or CD8. That is a decision that gets made in the thymus during development. Okay, so the basic review and kind of a compare and contrast for the two structures of MHC1 and MHC2. They look really, really similar. The main difference is what happens in MHC1. You have the beta-2 microglobulin, which is, again, not anchored in the cell membrane. It's free-floating and just associates via non-covalent interactions with the alpha chain for MHC1. For MHC2, you have an alpha chain that's 2 domains instead of 3, and a beta chain that's 2 domains instead of the beta-2 microglobulin. Both of these are membrane anchored. However, they both form very similar secondary and tertiary levels of structure. So you can see all these regions of spirally alpha helix in here, and the arrows tell you where the beta sheets are and kind of what direction they point. And you can physically see when you look at these proteins side by side, that structurally, they're virtually identical. Like, it's very hard to tell the difference between these. Yeah, this little loop and a couple of these regions are a little bit different in terms of their shape. The beta-2 microglobulin actually looks like it's a lot more beta sheet, and it's a little more compact compared to the alpha-2 domain, but they're structurally very, very similar proteins. Both of them create these grooves, and you'll notice that there's a slight, this is a hard to see difference in this, but there's a slight difference in the spacing. This one's actually a little bit more open on the MHC2 side, and that allows a little bit more space at the ends so you can get overhang of your peptide. Everybody feeling okay about the structure of MHC so far? All that protein structure, primary, secondary, tertiary, quaternary structure that I talked about in cell biology, this is where that comes back to haunt you a lot. Protein structures are fun. I like them.
They're also straightforward, which is nice.
They're also fairly straightforward. Okay, so again, kind of just a zoomed in version as a refresher again. I don't remember why I left this slide in because I've already talked about it ad nauseum. So.
It's kind of cool because you can see the MHC2 is noticeably bigger, so it can fit a bigger amino acid.
There's also a little bit of a difference if you actually look at the way these are drawn. There's a little bit of a difference in the depth of the cleft. So MHC1 has a slightly deeper binding groove compared to MHC2. And ultimately, these differences in those protein structures lead into how these different peptides can actually interact and bind in those grooves. So that's what this is showing. You've got your regions of alpha helix that forms the walls of the channel on MHC1, and you've got the floor that's the beta sheet of MHC1. You'll notice that this peptide is a little bit shorter than the one that you see for MHC2, because MHC2 overlaps more on the ends and isn't as clipped. It's also, this one is a little bit, it's narrower and deeper, so the groove is more like, I guess like that shaped as opposed to like that shaped. Slight differences in protein shape. And they have a huge difference in terms of protein function. One of those can bind to CD8, the other one binds to CD4. And they activate two different subsets of T cells. So what happens when we try to fit 9 amino acids into this MHC1 binding groove is usually there's certain anchor residues. This is the important part. In particular, amino acid number 2 and amino acid numbers 8 and 9. These are usually hydrophobic. This is what allows these to, and it's any hydrophobic amino acid. There were 6 of them, 6 nonpolar amino acids from cell biology. I don't expect you to remember what all of them were at the moment, and that's perfectly fine. But it's these two positions that sort of act as the anchors and sort of directly interact with the MHC protein. The rest of this is kind of allowed to sort of bubble out of that binding groove, and it creates these sort of loops. And depending on how long the fragment length is, whether it's 8 or 9 or even 10 amino acids, you may have slightly bigger or slightly flatter lengths. In comparison, MHC2, because it's got that more open binding groove, the peptides tend to lay pretty flat in that. It's shallower, but it's a little bit longer and it's open on the ends, so the peptides just kind of lay there and just drape and hang over the edges.
Did you say the anchoring were hydrophobic or hydrophilic?
Hydrophobic.
Hydrophobic, okay.
So the anchoring.
The anchoring amino acids are hydrophobic. Yes.
With this structure right here, the NC terminus that we're looking at here, this is the antigen, yes?
That is the antigen, yes.
Are there any secondary structures present with this level of antigen, or is it just the primary structure?
You might have some limited little bits of alpha helix left, but these pieces are really too short to get usually a full turn of an alpha helix takes about 7 amino acids. You're pushing it real hard.
Okay.
So you're really looking for primary structure.
So we can just recognize an antigen based off the primary structure of amino acids?
Yes, we can.
Wow, that's kind of crazy.
I have a chemistry question. How does being hydrophobic make it anchor?
Hydrophobic interactions with specific amino acids in the MHC protein itself will interact better with the hydrophobic amino acids from the antigen. That also promotes a tighter bonding, so there's not space for water in between those, so water molecules get preferentially excluded, and that means you get tighter association and it's not covalently bound in there. I mean, you can get these peptides to disassociate from MHC and float off, but it's uncommon. And the more hydrophobic they are, the less likely that is to happen. Okay. So, the MHC class 1 and class 2 molecules have some major differences in that peptide binding region. And again, class 1 presents peptides to CD8 positive T cells. That's cytotoxic TC cells. I'll add in the C's there. MHC2 does CD4 positive T helper cells. There's a difference in the peptide length that you can fit in those. 8 to 10 for MHC1 because it's closed on the end. 13 to 18 on MHC2 because it's open on the ends. So there are some very specific amino acids that actually anchor the peptide into the groove of MHC1. They are hydrophobic, which is similar to what we see in MHC2. But for MHC2, 7 to 10 of those amino acids out of that whole antigen need to be basically hydrophobic. Which, when I looked at that and I tried to do the mental math, I'm like, 7 out of 13. That's the majority of those little fragments. They need to be the most hydrophobic regions of those proteins. Yes.
Could you give an example of like what sort of peptide you'd find in the endogenous intracellular proteins? Like what would
Endogenous
or
intracellular proteins.
Endogenous?
Yep.
What does that mean? Like what type of protein are we talking about?
Okay. Endogenous proteins are proteins that come from inside the cell. We're talking about self proteins, normal proteins that are made by the cell, say little bits of the cytoskeleton or myosin or something like that.
Okay.
Or it could be viral proteins if your cell is infected. Exogenous proteins come from outside the cell. Normally, these would be little bits of things that your cell acquires through endocytosis, which may be little bits of bacterial proteins, for example. If you've got a bacterial infection, as your non-specific immune system destroys some of those bacteria, little bits of those peptides and pieces of the cell wall and bacterial proteins will get picked up by surrounding cells nearby. Okay, genetically, there are several hundred different allelic variants in humans. So genetically, we are pretty diverse with regard to MHC. I have a different kind than probably any of you do, meaning I would not be able to donate my tissues to you in an emergency. Sorry, no, not giving you a kidney. Love you lots. Actually, even between family members, though, it's very difficult to get a good enough tissue match that the immune system can ignore the transplanted tissue. So usually you have to go with someone outside even your immediate family. And that's because we actually express up to 6 class 1 molecules per person. So you're going to have probably 6 different kinds of MHC1 on every single cell of your body. MHC1 is expressed on all cells.
But you can have similar ones on the same cell, right?
So you can have 6 different kinds of MHC all on the same cell.
But can you have the same ones? So
Yeah, you're going to have like dozens to hundreds of copies of the same MHC1 on that same cell. And then MHC class 2, this was only expressed on antigen-presenting cells, APCs. You have about 12 different versions of that one that you're going to express on your antigen-presenting cells. So 12 different binding possibilities to recognize, let's just keep the SARS-CoV spike protein, call it 100 amino acids, with up to 10 plus domains that are little peptide fragments that you could recognize. Yeah, our immune systems can actually present tons of different kinds of antigens, and so can our native cells. We do this all the time. Every cell in your body presents some of its own self-proteins and basically says, "Hi, I am part of Dr. Steele. Please do not attack me." This is the basis for immune tolerance and ignoring your own tissues. Whereas the antigen-presenting cells that are specifically going to work with that are specifically going to have MHC class 2, those interact with CD4 positive T cells. So a single cell can have up to 10 to the 5th, 10,000 to 50,000 copies of these 6 different MHC1s or 12 different MHC2s. There's literally thousands of copies of the same MHC, but you've got 6 different versions of those, and they can all pick up little different bits of the same protein and display it. giving your immune system a good chance to find a fit with one of those bajillions of T cells that you made when you did your VDJ recombination from the last chapter. So numbers on top of numbers. The number of T cell receptors that you could make was gigantic, like incomprehensibly big numbers. For the B cells, it was somewhere up around 10 to the 14th to 10 to the 23rd different kinds. Your MHC basically also has to be able to accommodate similar numbers of antigens. But these are invariant. They don't change. They don't go through genetic recombination. Oh, thank God. You make the same kind of MHC1 or MHC2 your whole lifetime. And you inherit the genes from those from your parents. So while I might actually present a small segment from the N-terminal end of the SARS-CoV protein, you all might represent or present some of the fragments from the middle or even the C-terminal end of that same protein. It just depends on what parts of it my immune system can recognize the best versus what parts of it your immune systems can recognize the best. Question. Yes, Manal.
If we get infected, do we make a new type of MHC?
Nope, you make the same types of MHC all the time, period.
Okay.
They are set. So you're going to keep making the same 6 MHC1s your whole entire life. They do not change just because you have an infection. MHC2 does change in response to infection. So it will actually increase on the surface of your antigen presenting cells when an infection is detected. You'll start making actually more of them, but not different types, just more of the same type. You got 12 of them approximately, so that's plenty.
So we can only recognize a certain chunk of, for example, like a spike protein.
Right, we can only recognize a certain chunk of a spike protein.
So is it going to go through each little chunk and present it to the MHC?
Yes.
Okay. Until it finds
You
it.
will create tons and tons of different little chunks of those same proteins, and hopefully one of them is identifiable to one of your T cells.
So how close do we have to get for a transplant? How close are we talking? Does it need to be exact?
It does not need to be exact anymore because we also provide immunosuppressive drugs if it's not an exact match.
Did it used to be?
It used to have to be an exact match. This is why transplant lists are so long. This is why they encourage people to get registered on bone marrow donor registries, is so that if a kid has leukemia and needs a bone marrow transplant, because the probability of finding an exact match for your set of 6 MHC alleles on class 1 and 12 MHC alleles on class 2 out of the hundreds of possibilities is virtually zero. You're talking about 1 or 2 other people in the world who probably match your immunology genetics exactly. Technically, that's immunogenetics. There's a nice big word for the day.
Okay, I have a quick question. So my friend had a heart transplant, right? And the first one didn't take, her body rejected it, right? So she had to get another one. Why would her body reject it if she already has the immunosuppressant?
Because it's not perfect. No immunosuppressant gives you 100% coverage. You would have no functioning immune system at all, and you would be susceptible to every strain of bacteria, virus, fungus, everything. You wouldn't survive. You would have to literally live the rest of your life in a bubble. No food for you. No air. Can't have air. That might have pollen in it, or bacteria, or viruses. Water's dangerous too. Yes.
I noticed a lot of the monoclonal I picked and a lot of the other ones, they were doing that with the organ transplants with immunosuppressant. Do they kind of try to feather those where they bring it down enough where you don't react but still have a strong enough immune system to fight
Yes,
off?
that's exactly what they do. They tailor the dose usually to try to make it so that you're healthy and functional, but your immune system is not causing much, if any, damage to the transplanted organ. So it is tailored to you.
Okay, thanks.
Okay. So what you're looking at in this particular figure are just some of the examples from MHC class 1 variant 1 versus variant 2. And these are different amino acid sequences that are recognized by this particular variant. So at positions 2 and 3, what you actually have that promotes binding to that MHC class 1, I said position 2 was particularly important. That one needs to be a glycine, usually followed by a proline. And then down here, the other anchor amino acid is usually a leucine. That's what L stands for. For other versions of MHC class 1, that second position may not be a glycine, it might be a tyrosine. Still hydrophobic, nonpolar, or an isoleucine or valine. But both of these are basically nonpolar, any of these are all nonpolar, but those two positions in particular, position number 2 and position number 9, are the most critical for getting your antigen to stick into that MHC groove.
So is the proline in number 3, is that to prevent you from getting like a secondary structure or something like that? Or?
That's probably just an artifact of the way the floor and the walls of the MHC1 are shaped.
Okay.
So you have the smallest amino acid, glycine, and one that creates a 90-degree turn, usually in the protein. So that probably is also partially what causes it to like arch away from the floor of the MHC bed.
Cool.
Y'all have lots of good questions about this stuff. All right, these regions of identification that you see here are called epitopes. Singular epitope, E-P-I-T-O-P-E. That term is important. What an epitope is basically related to your antigen. It is the fragment of your whole antigen that is presented in your MHC. The chunk. Fragment or chunk of your antigen. AG, common abbreviation for antigen, makes writing your notes a lot faster because it's a term we use constantly. There have been more than 2,000 distinct peptides that were found on a combination of just 2 MHC1 molecules. So this is nowhere near an exhaustive list. Your MHC1 can bind, and you've got 6 of them, you can bind way more. So that's like that's like 6,000 different options that you could put there. That seems to be plenty. And that's 6,000 options for one protein. One spike protein from one COVID, one flu protein, one bacterial protein. There weren't even 6,000 possibilities if you looked at the number of possible combinations of like 10 amino acids from a 100 amino acid length protein. This works out to roughly what you're going to be seeing is anywhere between 100 copies of the exact same thing up to 400 or 4,000 copies per cell. And even one is enough to activate a T cell. You only need one. But there's going to be at least 100 and maybe up to 4,000 of them anyway, just in case, because why go kill when you can go overkill? The whole goal here is to make sure that you get interaction between your antigen-presenting cells and your T cells that are sufficient to turn on your T cells if that's appropriate. there's a reason we think that so many copies of this might be on the surface of our cells. So if we think about MHC1 specifically, that is supposed to present your self-peptides, which means you're presenting 6,000 to 60,000 copies of self-proteins. This is an awful lot of information for your immune system to have to sort through, but it really promotes self-tolerance. The ability of your T cells to not bind to those too tightly was trained in the thymus. So when they get out into your circulation, these help maintain those non-reactive T cells. They help keep them quiet, all those normal proteins that you display. When you start displaying abnormal proteins, you're still going to have at least 400 copies of the abnormal proteins, and so there's still a really good chance your T cells will pick up on that. But 400 out of 60,000 is still a relatively small percentage. Okay. Structural differences. The peptide binding domain. In class 1 molecules, that's made of a combination of the alpha 1 and alpha 2 domains of your alpha chain. Beta 2 microglobulin is not involved in forming the peptide binding domain. That is different than what you see in MHC class 2, where it's formed by both the alpha and beta chains, the groove for binding your peptide. On MHC1, it's closed at both ends, which is why it only fits 8 to 10 amino acids. On MHC2, it's open on both ends, which is why it fits a little bit longer stuff of 13 to 18 amino acids. In terms of what anchors those peptides, those epitopes into those MHC molecules, for the class 1, again, you've got these particular, usually hydrophobic amino acids at both ends, position 2 and position 9. For the class 2 molecules, those hydrophobic amino acids are usually distributed along the length of the whole peptide. Any hydrophobic seems to do. Like, it's not super picky. It can't afford to be picky because there's a ton of different pathogens out there. And again, for MHC1, the peptide epitopes can actually arch up and away from the center of that MHC groove and get more toward the surface. That's actually thought to promote binding and interaction with CD8. It should make it a little bit easier for the CD8 molecule to have its complementary match with that antigen at the surface of the cell. We're doing on time. Okay. All right. So, organization and inheritance of the genetics. You may or may not have had genetics yet, but you probably have enough knowledge of genetics to be able to follow this regardless because, well, you should understand basic Mendelian genetics by the time you get to this class. I know they're covered in intro biology, so everybody has had at least some basics and understands things like heterozygous, right? Okay. You remember how you had to do those Punnett squares, and one Punnett square for one gene was really easy because it's only a little 4-block table thing? And you liked those? Those were fun. Then you had the 2 gene ones, and they had 16 blocks, and you didn't like them anymore? Now imagine one with 6 genes.
Oh my god.
That's a big square.
That's a big Punnett square. And you're going to be heterozygous at all of them. Yeah.
For the possible genes for that, how many different genotypes could you get?
There are hundreds.
Cool.
Very large number.
Let's do it by hand.
Sit there and talk.
No, let's not do it by hand. There are some things where even with my level of love for this stuff, I'm like, no, I don't want to do that by hand. Could I? Yes. How long would it take me? A weekend, realistically, and a really big whiteboard. Like, I would have to take this thing home with me. Okay. So, class 1 MHC, this protein is actually a glycoprotein, which means it actually has sugars attached to it. It's expressed on pretty much all of your cells, and it presents endogenous or internal antigens to CD8 positive T cells. Are you getting this connection between CD8 and MHC1 and CD4 and MHC2? Good. The other major difference there is that CD4 and class 2 MHC are involved in detecting exogenous or external antigens. And internal and external, that really means with respect to the cell. Does it come from within the cell or does it come from outside the cell? There is a third class of MHC genes. They are not involved in antigen presentation, so we do not care about them. Yay. In fact, this is where your complement proteins come from. They are actually class 3 MHC.
Oh.
Neat, huh? There's also another one, TNF, tumor necrosis factor, and lymphotoxin are also encoded in the MHC3 cluster. Okay, so based on this, in mice and humans, these genetics are encoded very slightly differently. Again, I don't care about this green block because that's MHC3, and we don't care about those in this chapter. We're not going to worry about it other than noting that nifty, it's complement and a couple cytokines. Cool. Moving on. In mice, your class 1 genes are actually broken up into 2 different blocks. So there's been a little bit of a genetic recombination that has happened here that doesn't normally happen. It's unique to mice. In humans, you've got your class 1 genes, your class 3 group, and your class 2 group. You'll notice that you've got multiple options. So class 1, you've got an option for, you can make H2D, H2L, or H2K. And then you've got alpha chains. Those are just the alpha chains, because MHC1 has an alpha chain and a beta-2 microglobulin. Beta-2 microglobulin is not included for MHC1 because that's encoded on a completely separate chromosome in a totally other place, outside the MHC cluster. Why? I don't know. That's the way it happened, okay? Go ask somebody in immunogenetics. They'll be more fascinated by that than I was. I was happy with the just knowing it wasn't. So these are your alpha chains. That means those 3 domain pieces that form that L-shaped molecule that actually is important for making that MHC binding group. For MHC class 2, you've got H2A and H2E, and both the alpha and beta chains are encoded in this cluster for MHC2. It's a little bit different for humans, but pretty similar. So we're going to make HLA-A, human leukocyte antigen A, B, and C. Again, alpha chains, beta-2 microglobulins, still on a separate chromosome. And for our MHC2, we've got HLA-DP, DQ, and DR. Every human being alive today is pretty much heterozygous at all of these gene structure spots. So this is why you express 6 different kinds of MHC1. You express your mom's HLA-A and your dad's HLA-A. You express your mom's HLA-B and your dad's HLA-B. You express your mom's HLA-C and your dad's HLA-C. The probability that 2 other human beings got together and had a baby and had exactly the same types of MHC that created your combination approaches 0. Thus, tissue typing is difficult. And because you've got both alpha and beta chains that are involved in class 2, this is why you have 12 different combinations for class 2. Your mom's HLA-DP alpha, your mom's HLA and your dad's HLA-DP alpha, mom's HLA-DP beta, and dad's HLA-DP beta. Like, you've got all of these combinations that you have to make. And all of those are now your genetics.
Okay, I have a question. Let me out the box though. So like, what if they are the same? Is variation needed in order for it to function properly?
What if they are the same, and is variation needed for it to function properly? Yes, variation is critically important. Because the more variation you have, the more types of things you can recognize. If you actually have organisms that are homozygous at these genetics, you tend to have animals that have immune deficiencies. Cheetahs are actually the best example of this. The cheetahs that you see in zoos are inbred, we know this, and they actually have very restricted MHC, so their tissue types are virtually identical. In fact, you can transplant organs and tissues between zoo cheetahs without a lot of difficulty, which is uncommon, and that's how it was first observed. However, they're also susceptible to some infections, and because their immune systems are all virtually clones of each other, if one cheetah is susceptible to a particular, say, respiratory virus, all of them with the same genetics will be susceptible to that same respiratory virus.
Why do they allow them to inbred? Doesn't that now
Because we thought it was cool to keep cheetahs in zoos. That's why they're inbred. If you actually look at wild cheetah populations, you don't see the same problem.
Can you just introduce new cheetah genetics?
The problem with introducing new cheetah genetics is that the cheetah population worldwide is too small and doesn't support a lot of variation at this point. We can't breed back in more variation. Once it's gone, it's gone.
Well, that sucks.
Yep.
And this does happen, the homozygous.
Humans are rarely homozygous.
If they are, is it disease or is it just?
If they are, it's probably just at one of these genes so maybe like say your mom and your dad both actually had the same HLA-A, that's cool. But the probability that they had the same exact HLA-A, B, and C out of the 7.5 billion people on this earth is tiny. It's not zero, but it's a tiny probability.
So to get a transplant, you'd have to get an exact match of HLA-A, HLA-C, HLA-B. Also,
DP, DQ, and DR.
and also 3.
Because class 3 is not involved in antigen presentation, it does not matter.
Oh, so that's still okay.
Yeah,
I
that's
see the problem.
yes. It's a numbers problem, and it's a big number. Like, I don't like numbers that big.
Does inbreeding work on that, like in humans?
Inbreeding would work on that in humans as well. So we do see some just genetically distinct differences in some subpopulations of humans, like the Ashkenazi Jewish population, which is historically inbred. There's a number of genetic diseases associated with that, and there's also some pretty predictable autoimmune types of issues and susceptibilities to other types of viruses. In fact, I think in the early days of COVID, there was some hypotheses that if you were from some certain subpopulations that had a history of inbreeding, that you'd be more likely to be susceptible. I might be misremembering that. And that may have turned out to not be the case with further information. We've now had like 3 years to study COVID, so.
I have one more last question. Okay, so say, I just blanked. Oh, in order for a transplant to be successful, how many
MHCs have to match? As many
Yes.
as possible.
Okay. So
You're looking for the best possible set of matches for a transplant to be successful. The more mismatches you have, the more likely it is that you're going to have rejection.
So if I have one MHC that's matches, there's a chance that it still could work.
If you have just one MHC that matches, there's almost no chance that it'll work because the others are too different.
Do you almost die from the rejection?
You can.
Yeah.
Tissue rejection can kill you.
It's pretty.
And it's not just that it's going to destroy the organ. The whole process of runaway inflammation that takes place in the bloodstream and everything else causes a lot of damage throughout the body. But yes, it'll actually kill you.
graft versus
Aside from the fact that you're literally attacking the heart that you kind of need for functioning, or lungs or whatever.
Do identical twins have the same exact?
Identical twins do have the same exact MHC.
So that's why they do say it's like a walking bag of organs.
So yes, it is a walking bag of organs for you if you have an identical twin. That's also why cloning technology is so intriguing to us as humans, because that means that we could actually take our own MHC genetics And if we could grow up like, oh, say just like, I don't know, a kidney in a petri dish, that would, from cloned cells, that would be awesome because it would be a perfect match for our MHC and there'd be zero risk of tissue rejection because it is literally from our bodies.
So, I know some organ transplants don't require like a perfect match at all, and you only care about blood type.
Kidneys are notoriously like somehow less susceptible to attack by the immune system for a mismatch of MHC.
Do you have any idea as to why?
Short answer, partially.
Does the immunology
Long answer,
community
it's
know?
complicated.
Does the immunology community as a whole know, or kind of?
I have not actually spent any time researching why kidneys in particular only require that you have a blood type match. However, what I will say is that if you get a kidney transplant and it's only based on blood type matching and you're a poor match at your MHC genes, you'll still be on immunosuppressive drugs.
Okay.
Yep.
Good to know.
Yeah. Okay. So this is a pretty simplified map of the mouse and human MHC gene regions. Remember that the class 1 genes were split up. So you've got K, D, and L. And then you've also got the class 2 region here in blue and all of these genetic types that may exist in there. So there's multiple alleles that you can have for these genes. You're not limited to just a simple Mendelian trait that has one allele. You can have hundreds of different versions of this gene. So it's not just, to put it in simpler terms, it's not just brown eyes or blue eyes, it's every shade of eye color on the spectrum. Your immune system isn't just A or B. It's everything from, we don't even have that many letters in the alphabet because there's literally hundreds. So A to Z, and that 5 times over. So it's an absolutely gigantic number. It really is. Again, for mice, that cluster 1 is split. For humans, that cluster 1, MHC1 cluster remains as an MHC1 cluster.
Lucy or Loki?
What?
Is it Lucy or Loki?
Loci.
Loci.
Locus is singular. Locus, this is a term that got thrown around in genetics all the time that nobody bothered to define for me properly. A locus is a region on a chromosome. It's just a physical location of a gene. And I'm going to abbreviate chromosome because I ran out of space.
So in normal human genetics, allele is just 2 copies, right?
In normal, for a simple Mendelian trait in humans, there's only 2 copies of a particular allele. So you would either have like brown eyes or blue eyes, or light skin or dark skin.
So we have many alleles?
But we have many alleles. We do not have just 2. And skin is actually a really good example of that. We are not just dark or light. We are every shade in between because we have multiple alleles. In mice, if you look at what contributes to the coat colors and patterns of their fur, there's over 16 genes involved in what makes a mouse look like that mouse. Like just its physical outward appearance, from the color and texture of its fur to whether it has super soft, silky fur or what, but there's like 16 different genes involved. So if it's that complicated for mice, it's just as complicated for humans, which is why we have so much variation in hair color and texture and patterns and curl patterns and skin tones and all of it. It's because very few of our genetics really are simple Mendelian traits. We teach the simple Mendelian traits first because it's easy to understand the 4 square Punnett square. And then we tell you it's really much more complicated than that. And here's this, 2 allele version that you could have. Have fun.
I have one question. Are there any cells I don't have or that don't involve MHC?
There are cells in your body that express less MHC than other cells in your body, but they all express MHC1.
Okay.
MHC2 is pretty much not expressed on most of the cells of your body. It can be if there's an infection nearby or in the cell, but it's usually at very low levels compared to on antigen-presenting cells, macrophages or dendritic cells. Good question. Okay, these genetics are inherited as a block. They are physically so close together on the chromosome that they're considered linked genes. And if you've taken genetics, you know what that means. And if you haven't taken genetics in a while, it's literally linked genes just means that they're physically so close that they tend to get inherited all as a set instead of getting separated during recombination. So usually you get a haplotype. You get one haplotype from each parent. You'll get your exact same mom's set of MHC A, B, and C, and for MHC1. We've actually taken advantage of this in mice, and we've created inbred mouse strains specifically on purpose so that we can study how the immune systems work. And there's two terms that we tend to use with regard to mice in particular, but it also kind of applies to humans. Syngeneic means identical at all genetic loci, gene regions. Unless you spend hundreds of generations inbreeding your mice, this is difficult or impossible. We do have mouse strains that have been inbred for that long. Congenic mice are genetically very similar. They're identical except at a single genetic region, one place that's different. So any differences between those two mouse strains have to be due to that one genetic difference. And one of the first places we did this was in immunology. Humans, there are 7.8, last I heard, 7.8 billion people on this planet. We are genetically outbred. We are not inbred. Some populations, even the most inbred populations that you can think of, are still relatively outbred compared to like these lab mice strains. People marry into families and out of families all the time. But things like religion and politics play a role. In genetics, I know that you talk about like what happened in the Russian royal family with regard to hemophilia and other genetic inbreeding things that happened because that's how you hang on to your wealth and power historically. Is you marry your cousin or your uncle if you watch Game of Thrones. I wouldn't know anything about that. I don't get to watch a lot of TV anymore. Somebody told me I would like Game of Thrones. I was unprepared. These genetics are also codominant. So again, it doesn't follow simple Mendelian dominant or recessive. These are codominant, which means you express all of them simultaneously. The body does not care. It doesn't have a preference or a bias toward one type of MHC or another. It's just going to put them both up there like having type AB blood. You're going to have type A, B, C, D, E, F, MHC. That's a bad alphabet salad. I should space that out more. So ultimately, because you've got both maternal and paternal MHC genes expressed in the offspring, this gives us the best chance to have some capability of presenting all the possible types of antigens that you can encounter in your lifetime. Lots and lots of possible combinations, hundreds of alleles. You're going to express some subset of 6 of those out of hundreds. And each of those can accommodate a variety of different types of peptides, like up to 2,000. So this makes transplantation difficult because humans are basically heterozygous at each and every locus. Non-matching MHC patterns results in rejection of transplanted tissues that we've actually already talked about. And these characteristics, again, were originally noticed in inbred mouse strains, and we took advantage of that. So if you like the graphical versions of inheritance, which I personally definitely do because it helps me to understand, if you start off with a homozygous mouse of 2 different types and you breed those 2 mice together, you get a heterozygous mouse.
Cool.
that expresses both. This mouse is a little bit more outbred than its inbred parents were. If you try to do tissue transplants, so they try to take a piece of skin. If you take a piece of skin from a BB parent, you can transplant that to a BB to a BB other mouse, or to a BK mouse, but not to a KK mouse, because the immune system will see that patch of skin and go, that is not part of me, its MHC does not match mine, it is foreign, and therefore I should destroy it. The same thing is true for KK parents. If you have a heterozygote, though, because it's codominant, because this mouse expresses both B and K, neither parental type of mouse can actually accept a transplant, only another heterozygous mouse could. And in human families, they've also got a graphic representation of that. So you've got a mother that, say, is expressing type A and type B, the father, or sorry, that's the father, type A and type B, the mother, type C and type D. And the potential combinations in offspring include AC, AD, BR, R wasn't even a thing up there, so I don't know where that came from, BC, and BD.
It's like a mix.
It is a mix.
What was that called?
Mutation.
Oh no, it looks like
Or
a
recombination.
translocation of like C and D to get R.
It might be a translocation of C and D, or it's recombinant, most likely, meaning it has features of both. Go ahead.
So I know that NK cells, they target cells with low levels of MHC1. Are they also looking to see if it's a different type of MHC1?
I don't know.
I'm just gonna figure that out. I don't know.
Okay.
This makes
So yes, as one of your classmates just commented, imagine showing this to someone random who has no idea what's going on. This would not make anything clearer. I have to agree. However, if you sit down with this figure for a few minutes and you actually kind of sort out what's going on, you do figure out that ultimately this is about how you can create different combinations of these different types of receptors. So up at the top, you've got the class 1 molecules, and at the bottom, you've got the class 2 molecules. The class 1 has the alpha chain, which does go through, or has 3 different genes that encode different versions of alpha chains scattered throughout your genome. So you can actually make the KK gene or the KD gene if you're heterozygous, or you can make the DK gene or the DD gene, or the LK gene or the LD gene, depending on which of these you express. And because these are codominant, you actually express all of them. So you have all 6 of those versions of MHC1 on the surfaces of your cells. And then the next chapter, we'll talk about why that's important, or at least a little bit about why that's important. But among other things, it's important during T-cell development. After the cells are developed and mature, it also plays a really, really critical role when you're actually talking about antigen presentation and trying to activate and turn on a naive T cell that has not yet encountered antigen. On the bottom, for the MHC2, you'll notice you've got actually 2 clusters of genes, the alpha beta or the E beta genes, and you've got the K haplotypes or the D haplotypes. So you can have an AK, AD, which is the pink and maroonish colored thing. So you've got various combinations of these. You can express just the pink. You can express the pink and the red. You can express the yellow and the orange, or just the yellow or just the orange, and that's where all of those combinations come from. And you can sort of sort through and figure out what happens for the alpha chain and the beta chain, which are encoded separately for MHC2. Yes.
so for like the leftmost one, right?
This one.
No, on the chart,
This
the KK and the KD for MHC1. Is that on a different chromosome than DK, DD?
No, it's in
Okay.
the same. It's on the same chromosome. It's in the same gene cluster. It's just separated a little bit.
Okay, and then
So it's still actually inherited as a block and linked.
all these are inherited.
All
as a
these are
block.
inherited as a block
It's
and
all
linked.
on the same chromosome in the same area.
Yes.
Oh,
It's all the
okay.
same chromosome, same area.
So I'm sorry. On this, which one of them is only for MHC1? Only
MHC1 is KK, DK, and LK.
no, like on the surface itself. All of these, only these. I mean, those are only for MHC1.
Those are MHC1, yes. What's different about MHC than what we talked about with the T cell receptor and B cell receptors is that MHC doesn't go through genetic recombination. So you don't have to worry about VDJ combinations or anything like that. These are just normal genetic genes. They're fixed. Whatever's in your genome is what you actually express. And again, you inherit that from your parents, and then you express the combination of both your mother's and your father's. And that makes it uniquely yours. And the reason I say uniquely yours is because of this next slide, which has a big long table in it with lots of different gene options. But this gives you an overview of the types. I said next slide. Okay, that's better. So on this slide, it actually shows you some of the options or how many types of proteins are available for each of the types of the MHCs, etc. So you've got the MHC class 1, A, B, C, E, F, and G. I know D is missing from that list. But for the A gene, there's 2,480, and you can express these in combinations with the 3,221 Bs or the 2,196 Cs. For E, F, and G, the numbers are much smaller, but you still get the general idea that this gives you a vast array of potential combinations. I actually added this up because I'm like a numbers person, and the sum total of allotypes for all of these is 10,900. That's a lot. So your chances of actually sharing an MHC match with somebody is less than 1 in 10,000. Of these, MHC1 had 7,927, MHC2 had 2,973. I don't think it's particularly important that you remember that. And that's only a fraction of the actual alleles that we have. These are just the ones that encoded functional proteins. There's actually over 15,000 alleles. It just turns out that about 5,000 of them are broken and don't actually encode anything functional. So pseudogenes mostly. Even with that, the genetic DNA differences is only maybe 5 to 10%, which sounds like a very small number, but in terms of genetics, considering we're like 98% the same as a mouse, we're all mammals, that's actually a lot of genetic difference in this particular region. So it doesn't seem like a huge amount of variation, but at a species level, 5 to 10% for every person, and there's 7.8-ish billion of us, that adds up to a lot of genetic variation across the whole human population. From an evolutionary perspective, because I used to teach evolution too, this is really pro-survival. It's useful to be able to recognize and present antigens on all kinds of different MHC molecules, basically kind of in the hopes that one of them works with one of your T cells. You'll have a set of 6 of these, and they'll get trained in the thymus to recognize your particular ones, but we'll talk about that more next chapter, 8.
So 2 quick things. The allotypes, are they amino acid sequences? Like
Yes.
Okay, so the amount of amino acid sequences, not the amount of like polypeptide chains.
Correct. That's not the number of polypeptide chains. That's actually the number of proteins that are in code. So it's basically like the number of genes that make functional proteins.
Okay. And then my second thing is, do you want us to know this chart?
No, do not know that chart. I am not going to ask you about that chart.
Cool.
Okay, as with the alpha and beta receptors, though, we do have regions that are a little bit more variable in the genome, but you don't see a big spike in any of the alpha 1, alpha 2, alpha 3 sections like you would kind of expect to. They're actually fairly consistent. You do have a little bit maybe like right there for alpha 1, But remember that the structure of this protein is different. So your alpha-1 domain and your alpha-2 domain actually form the walls of that MHC peptide binding groove that your antigen is going to fit into. It's a different system than the T cell receptor. And it doesn't bind in or interact in quite the same way. So it doesn't have those variable regions. So what you've got essentially is this pocket on your MHC that your antigen is going to fit into. And then you've got that T cell receptor we talked about, that it had those 3 regions in particular that were kind of hot spots in the variable region that sort of looped out a little bit farther away from the surface. And those were critically important for interacting with the antigen. They're the ones that sort of have to be able to reach into that pocket a little bit and actually contact the peptide that's going to be mounted in this binding groove. Most of the differences in amino acid sequences tend to be clustered within the groove sites. If we changed the areas outside of this too much, we would end up with a molecule that couldn't effectively present antigen to T cells. So evolutionarily, the shape is basically conserved, but you do see a few little areas here and there that allow some variability. But you've still got, for MHC1, you've got those 2 or 3 conserved amino acid residues at positions number 9 and number 2. And then for the MHC2, if we were looking at an MHC2 binding groove, it would be large or more likely to be hydrophobic in here. Ultimately, this is all about the protein structure equals function problem. So I know I say that in cell biology all over the place, structure equals function. It got drilled into me really, really hardcore in my biochemistry classes. Like, that was literally, my teacher said, if you don't know the answer to the question and you know nothing else, write down structure equals function, you'll get at least 1 point. Now, that was true for my biochem professor. It's probably not true for yours. But his tests were hard, so.
I'm trying to understand this point. It says that the differences is at the level of the amino acids, right? Okay.
Yes. What you're looking at is the amino acid
The
variability.
outside of the group.
Inside, within this, so these little spikes kind of correspond with these little hot spots that are sort of indicated in the protein
That's
structure.
where the changes happen, but not
That's where the
outside.
changes actually happen, but not many outside of that region. So this region here is pretty well conserved.
Okay.
And that's probably going to be most of where the peptide does, our antigen peptide does a lot of its binding. Is going to be mostly here and some out here.
So are there ever like chain or polymorphisms that happen with this that just make it not functional? So like if you swapped out, let's say like amino acid
Well,
7,
if you don't have functional MHC, you don't have really a functional immune system. So chances are you don't survive to adulthood. You could be born without functional MHC, but you're not going to survive your first infection.
that makes sense.
In the wild. With modern medicine, you might, but it still means that if you think about it, your lifespan is probably going to be much, much shorter if you are born without functional MHC. That means you can't present antigen to T cells. Your ability to deal with viruses and bacterial infections and all of that is limited to just what your innate immune system can handle, which is not everything. So that brings us kind of to trying to understand a little bit about the role of MHC molecules and what they're actually doing and why. Why is one of my favorite questions in all of science, and I ask this a lot, even when I'm reading things and trying to understand, because I want to make sure that I understand what's happening and why that particular system is in place or why it works this way. So why present antigens this way? There's a few different reasons. Using MHC to present proteins, and especially to present self-proteins, little bits of your own cells, the normal stuff. This is one way to demonstrate that your cell is normal and healthy. Like, there's just normally bits of proteins that your cell makes, like collagen and stuff, that get degraded on a regular basis, and little bits of them get stuck in these MHC molecules and stuck on the surface of the cell. And this is just one way that the T cells know to ignore that cell. It's just showing collagen. There's no threat there. It's no big deal. We can ignore that tissue and keep moving. But it also allows us to show specifically when a cell becomes infected. Those self-peptides change. and it changes what's in MHC1, but it can also change what's in MHC2. We haven't talked about those pathways yet and how those are different. So that's hopefully something that we get to today because we need to. Again, MHC1 is important for activating cytotoxic T cells. MHC2 is important for activating helper T cells. Displaying those self-peptides is also really important during lymphocyte maturation, so we'll see a lot of role for this when we look at the next chapter about T cell development. And it helps to maintain tolerance in secondary lymphoid organs. That means it helps keep the immune system quiet and not attacking our tissues. Apparently, our lymphocytes critically need to see bits of normal proteins, and if they don't see that, then things start getting kind of screwy.
So the MHC is usually they're just presenting their cell protein until there's an infection, then they go back and they present.
Yes, so MHC will present self-proteins until the cell becomes infected, if it ever becomes infected, at which point it will express a mixture of viral proteins and self-proteins. Because it doesn't stop making self-proteins, it's just a random sample of whatever's inside the cell. If that random sample happens to include some virus or bits of digested bacteria, then that triggers the immune system.
You said lymphocytes need to do this or else things go
awry.
What things
Like if the immune system didn't see these normal self-peptides, chances are good that it would start attacking our own tissues. That leads to autoimmune conditions. And we see a lot of health problems today that we're starting to understand that are autoimmune. Some of them we knew existed, like diabetes, but we didn't realize that it was actually type 1 diabetes is actually caused by the immune system. Type 1.5, 1.5 diabetes, you probably haven't heard of. Anybody? Oh, new thing. Yeah, there's a type 1.5 diabetes. It's actually intermediate between type 1 and type 2. Type 2 was always considered to be adult onset, except we're starting to see this more in children. Type 1.5 diabetes is autoimmune, but it takes effect as adults. So it's something that develops later in your life. Currently, I guess estimates are late 30s, mid to mid 40s is generally the range you start seeing that.
So it's just like it's expressed late.
It's not just that it's expressed late, it's that something goes wrong in the immune system training profile and that happens randomly, like when you get older. It could happen theoretically at any age. I'm going to bet that most of your doctors have never even heard of type 1.5 diabetes. I see some head shakes like, no, that's not a thing. Yeah, that's a thing. You'll find it in the medical literature and you'll find it like all over the internet, but it's doctors don't care because it doesn't really change the treatment plan that much. But what it does mean is that for type 1.5 diabetes, it's probably not because of your diet. And being able to control your diet is not going to be enough to get that to reverse or to effectively manage it. It means you're probably going to become insulin dependent, ultimately. Not through any fault of your own, just because your immune system screwed up and decided that your pancreas was actually a threat to your health. Oops. In Crohn's disease, your immune system gets screwed up and is convinced that your intestines are a threat to your existence. In multiple sclerosis, it thinks your immune system is dangerous. Like there's a whole host of things that go wrong, and the spectrum of how wrong varies from person to person and immune system to immune system. But all of it is because of some sort of failure in the ability of your immune system to detect normal healthy tissue versus your own tissue versus invading pathogens. Okay, because I'm trying to drill this into your heads very, very thoroughly and very, very completely. MHC class 1 presents intracellular antigen. This and peptides. This comes from within the cell and it includes self-proteins. This is a way of checking to make sure that cells are generally safe and healthy and belong to the organism. For us, that would mean you, your body. It can show which cells have been infected with viruses and are abnormal, or it can also show tumor cells that have become cancerous and that are abnormal. Either way, this is an abnormal cell. It's going to display those peptides on MHC class 1 and activate your cytotoxic T cells. Class 2 is a different system, and it's not expressed on every cell in the body. It's only expressed on professional antigen presenting cells. That includes macrophages, dendritic cells, and B cells, just to reiterate. Those are probably, hopefully, getting drilled into your head also. So these antigens usually come from outside the cell, external or exogenous antigen. And it helps the immune system to respond to threats against things that shouldn't be in our bodies, but that somehow are, but that are still not part of our own cells. So it tells the immune system, this threat is coming from an external source. Please go look for like a bacterium or a parasite. It's that type of thing as opposed to a viral infection of our own cells. Believe it or not, when I read through textbooks and stuff, like when I'm reading your chapters and it says professional antigen presenting cells, I actually stop in the middle of the paragraph or sentence that I'm reading and list them. Just for review purposes. Make sure that knowledge is fresh in my head too. Do I know it already? Yes. Do I do it anyway? Yes. Why? I don't know. I just do. It's part of how I study. It seems to work because it helps me integrate all of that knowledge, and the better integrated stuff is in my brain, the better it sticks. Okay. Professional antigen presenting cells. Dendritic cells, macrophages, B cells. I just said that. Other non-professional cells that can also present antigen include fibroblasts, like your skin cells, glial cells in your brain, and the pancreatic beta cells of your pancreas, the ones we were just talking about that are important to type 1 and type 1.5 diabetes. The thymic epithelial cells, we will talk about those little guys a lot in the next chapter. Intraepithelial lymphocytes, vascular endothelial cells, all of these can present antigen, but don't do so as with MHC2 unless there's a major infection. Like all of your cells have the ability to upregulate a little bit of MHC2, but we're talking like trace levels. Enough that your immune system is going to be looking at that going, that's so cute, I think you're mistaken, and ignoring, mostly. What it's going to be looking for is MHC class 2 on a cell that is specifically intended to do nothing but present antigen. That includes your professionals, the dendritic cells, macrophages, and B cells. Yes?
Do you see MHC1 on erythrocytes? Or?
MHC1 is sometimes still present on erythrocytes, yes.
Platelets, I'm guessing, not really.
Platelets, no, not really at that point. They're not really cells per se anymore, and they don't have any living components left in them, so they're not capable of processing and presenting antigen.
Gotcha.
They may have, I don't know, I guess residual MHC from when they were cells, but that would be about it.
Cool.
Let's see. So I guess the one thing I want to say about this is that the MHC expression profile changes as conditions in your body change. So I suppose right now I could, like, instead of using humans as an example, I could talk about kittens with really bad bone infections because I happen to have that going on, and there's a lot of that concern going on in my life right now. But in this particular case, it's bacterial rather than viral. So the patterns of MHC expression are a little bit different. His are going to look different than a normal healthy kitten's. Mine looks different than somebody who's infected with something, whether that's a bacterium or a virus. When you get an infection, your cells increase expression of MHC2. All cells already express buckets of MHC1. They don't really need to increase that, but they can, and many of them do start to express low levels of MHC2. It's basically kind of an early warning system or an activation marker is what one of the things we use it for. It's like saying, oh, look, that tissue, that one right there, that's the spot of the infection. See all that extra MHC2 that's not supposed to be there? Yeah, signs of infection. Viruses, however, like to screw you up really badly, and they tend to actually cause your cells to downregulate MHC1, so you start to express a little bit less of that. It's a really interesting cheat code for viruses. They're trying to evade being detected by the immune system. The best way to do that is to make sure that their little virus proteins don't get shown on the surface of the cell. If it's not shown, they're invisible, they've slipped under the radar, and that's evolutionarily advantageous for the virus. Bad for us, good for the virus. The good news is that you've got a backup system for that. So if you are infected with a virus that causes your cells to decrease their MHC1 expression, NK cells start to pick up on that. They're the ones that specifically go around and kind of check to make sure that levels of MHC1 are normal. Some cytokines also cause changes in MHC expression, and corticosteroids, so things like prednisone is probably the most common corticosteroid prescribed for humans anyway, and oftentimes for pets too. And some of the prostaglandins can actually decrease MHC expression as well. Your body's constantly kind of tweaking the levels of these things a little bit in response to cellular and tissue conditions. Exactly what's going on depends on what, if anything, is present in your system. Chances are good that you're all fighting off something right now and you don't know it. Like if there's anything you learn in immunology, it's basically we're under constant assault from all kinds of bad, icky pathogens. Microbiology drives this point home. They're all probably bacteria. If you take microbiology, like there's just bacteria everywhere. Then in this class, it's like, and viruses, don't forget the viruses. Now you have bacteria and viruses and parasites and fungi. You're gonna die. But that's why you have an immune system. Okay, so this different ability to present antigens can dictate overall the strength of the immune response from individual to individual. On the internet and talking to people, a lot of times you'll hear, you need to strengthen your immune system. Okay, how? Like this is a complex and intricate process. I'm not sure exactly what part of my immune system they're talking about strengthening, but the problem is that I've totally overanalyzed that question at that point. I can't exactly tell my body, you need to make more immune cells. There's infection running around in the world out there. Like it doesn't work like that. It just doesn't work like that. Drinking extra vitamin C does not strengthen your immune system. Taking extra supplements does not strengthen your immune system. Like there's a limited number of things that you can do to make sure that your immune system is functioning optimally. And probably the better ones are maintain a healthy lifestyle. So eat the healthy foods, get some regular exercise. It sucks on both fronts because it doesn't taste as good as junk food and nobody likes running. The people who do like running think they like running. Someday they will realize that they no longer like running. So their time is coming. I hate running. So there's a couple different issues though that do come up, and two explanations for variability in the immune responsiveness. Like, why do I get the flu and you not get the flu? Is it because my immune system was somehow weak? Maybe. Maybe there was just a hole in my immune system. Maybe my immune system didn't actually make a T cell receptor and an MHC pair that could actually function together to recognize that influenza. Oops, bad for my immune system. But like the chances of that are slim because the numbers of cells that you generate is very high. It's still possible. That's basically the holes in the repertoire model. T cells with T cell receptors that can recognize certain foreign antigens, and also the ones that look too much like your own antigens might actually just get ignored by your immune system. So let's say a virus comes up someday, a zombie-causing virus or something. Since we're getting close to Halloween, we will put in zombies now. Let's say the super future zombie virus actually kind of mimics your own tissue. It looks like it's supposed to be you. It looks like, say, normal collagen or something. How is your immune system supposed to tell that that's not normal collagen? It can't. It looks like normal collagen, it's going to treat it like normal collagen. Your immune system's basic strategy, it looks like a duck, it quacks like a duck, it's probably a duck. It looks like a normal U protein, it's probably a normal U protein, because that's what it got trained to recognize when it was in the thymus, was all of the U proteins. The other model of kind of things that happen here is the determinant selection model. This is when MHC class II molecules differ in their ability to bind particular processed antigen peptides. So maybe some of the MHCs can't really optimally bind all of the different parts of the flu virus or the zombie virus or E. coli bacteria, to give a few examples. The interesting thing is that both of these models have experimental evidence that supports them. So yes, there are holes and gaps in our immune systems, and that's unavoidable. There's nothing you can do about that because that's all genetically determined or randomly determined. And you have no influence over those processes. It doesn't matter what you eat. It doesn't matter what supplements you take. It doesn't matter if you drink the fancy black water that contains fulvic acid, minerals, and all of the elements that are in nature, which is terrifying. There's this stuff on the internet. I got introduced to it in another class. It's called black water. In their advertising, they say contains all the elements. I'm like, yes, I need more thorium in my diet and uranium and arsenic.
Does it have those like unstable
All?
ones?
Yeah,
The
including,
unstable ones
apparently
don't
it's
have, like,
supposed to have the francium and the lawrencium and all of the
Yeah.
really unstable ones too. And I'm like, this is not good.
Yeah, you put it in your body.
What?
It's very nasty.
It's actually not that bad. I bought a case of the stuff for Halloween.
How does it taste?
Like really soft water.
You should do
Like
column
it's
chromatography.
very slightly different.
You should do column chromatography on it. See what comes
I should do column chromatography on it.
Yeah. You never know. What if it had like a bunch of weird stuff in it? You'd only find
Then
out-
column chromatography would not be enough. I'd have to do HPLC, high performance liquid chromatography or gas chromatography.
Well, that's just too much work.
That's also expensive.
Column chromatography.
Okay. So our T cells are restricted to recognizing peptides that are presented on MHC. That means this is the only way they can really be activated is if they bind to a peptide that has been presented on its little MHC pedestal in that nice little binding groove. There's good reason for that. That serves as a signal that yes, this is actually an antigen and that yes, this is something that the T cell should recognize. And there's also co-stimulatory molecules that have to work with it. We talked about some of those back at the beginning of the semester, CD4, CD8, CD25-ish in there, 28, depending on what type of cell. There were all these co-stimulatory molecules that also had to provide signals. So all of that basically tells the T cell, yes, you're seeing this, yes, this is valid, and yes, please respond. You need a whole bunch of yes signals before your immune system is supposed to kill things, because ultimately what cytotoxic CD8 T cells kill is parts of you. It's going to go after and kill your own cells because those cells are infected. You want them dead so that they don't continue infecting the rest of your body. But you don't want your immune system killing you all the time. That's autoimmunity, and that's bad. So we also know that the MHC haplotype, the genetics of the MHC actually have to match the haplotype of the T cell. They did some really interesting, elegant experiments, and I don't know about you guys, but remembering particular experiments that show particular things is not a specialty of mine, nor is it overwhelmingly my primary interest. I want to know how things work and not necessarily how it was demonstrated, so I kind of just tend to skip over those. Also, less things you have to memorize for the exam, because I don't care if you remember that it was Rolf Zinkernagel that actually determined this. Like, that's not the important part. The important part is that you have to have a genetic match. There were actually 2 experimental models that showed this, so we are pretty strongly confirmed that this actually is a thing and that it is important. The antigen-specific proliferation, so when you see an antigen and the T cells recognize it, one of the things they're supposed to do is make tons of copies of themselves and go through mitosis a bunch and expand. That only happens when the macrophages or the dendritic cells matched the same MHC2 haplotype as the T helper cell that could recognize the antigen. Similarly, For CD8 and MHC1, they found the same thing. The genetics have to match. Again, CD8 T cells recognize MHC1, CD4 T cells recognize MHC2. Do y'all have that ingrained into your memories permanently yet?
It was on TikTok yesterday.
It was on TikTok?
No, I'm not joking, because I'm on like.
But you have to send me the TikToks of the immunologies.
It was like a way to remember it is like the number for the CD4 multiplied by the number of the MHC always has to equal 8. So like CD4 and MHC2, 4 times 2 is 8. So you remember which ones go together.
It's actually pretty good.
That actually is pretty good. 8 is
also
also the magic number of electrons in the outer shell of an element, so.
It's just the magic number.
8 is a magic number.
I'll send it to you.
I thought 7 was the magic.
No, 7 is not the magic number.
Sounds like a good number for halogen.
Okay. So T cells have to see these peptides, little fragments of proteins presented in these MHCs. Whole antigens are too big. Remember, you had to trim these things down somehow, and this is the part that we hadn't talked about yet. You actually have to trim these proteins down from their full protein length of hundreds or maybe even thousands of amino acids into a fragment that is 9 for MHC1 and like 13 for MHC2. Tiny bits and pieces. So your cells somehow have to process those antigens first and degrade them. Chop them up into little bitty bits. Those small protein bits, those are known as peptides. Only the processed antigen peptide fragments can be recognized. Your immune system cannot recognize a full-length protein. It ain't that smart. It's too big. Like, it would be too physically large and too physically clunky to try to do all of that. Being able to recognize little bits and pieces of it that are hopefully unique to that particular pathogen, that would be good. So there's two different pathways, depending on whether we're talking about MHC class 1 or MHC class 2. And these two different pathways arise because these antigens come from two different sources. One comes from within the cell for MHC class 1. The other comes from outside the cell for MHC class 2. So class 1 presentation requires what's called cytosolic or endogenous processing. And that's the pathway on the left over here. You have a protein that is from within the cell, inside the cell itself, just floating around in your cytoplasm. Those get degraded by proteasomes. Hopefully you remember a little bit about that from like intro biology. I don't spend any time talking about it in cell bio, so here we go.
Proteasomes are the lysosomes?
Proteasomes are similar to lysosomes. They're not acid-based. A proteasome is basically, I think of it as like a drum that you stick your proteins in, and it's got enzyme activity, and it cuts up your proteins, and then little bits of proteins come out. Since it's a protein blender. That's a very good analogy. I really like that one. Yes. So your protein goes into the proteasome protein blender, little bits and fragments come out. Those little bits and fragments then get sent into the rough endoplasmic reticulum. And that's where they actually get associated with class 1 MHC, is in the rough ER. So in cell biology, we do talk about this part. The rough ER is part of where protein translation sort of occurs. So the ribosome kind of docks on the surface of the endoplasmic reticulum and squirts the protein into the lumen of the endoplasmic reticulum. Once it's in the ER, that's where it gets like sugars and stuff added to it, and then it goes to the Golgi. Vague memories of some of that, a little bit. Sounds basically familiar. That's the basic summary of what happens in there. So in this particular case, some of those peptides, some of those protein fragments get associated with your MHC class 1. Which ones? Yes, All of them, many of them, a random 50,000 approximately piece assortment of random sample of all the proteins inside your cell. You got about 50,000 MHC1s on the surface of your cell, so that's about 50,000 options that you can choose from. And they're constantly turning over. So if you miss one this round, it's okay, you'll get it the next round. Your cell will present all of these, or many of these, at some point or another. Once that little antigen fragment has been loaded onto MHC class 1, it gets exported through the Golgi, just like a regular protein would, sent to the surface of the cell, where it will display that in its lovely little MHC binding groove. MHC class 2 proteins come from outside the cell. So their pathway to incorporation is a little bit different. It doesn't happen in the rough endoplasmic reticulum. The protein MHC class 2 still gets made and it enters into the rough ER, but it doesn't get its antigen back here. It actually starts traveling through the Golgi system, heading toward the surface of the cell, and then later on in the pathway is when it gets its antigen. So in this particular case, as the cell takes up antigens from their environments, and again, this is specific for antigen-presenting cells. Dendritic cells in particular are the best at this. They do this consistently. So they're constantly sampling their environments, eating and drinking little bits of the extracellular fluids and substances that are around. So maybe that's a little bit of collagen or something, or maybe that's a little bit of a virus. Either way, the antigen-presenting cell eats a little bit of that, digests it in a lysosome, chews it up, probably gets a little bit of additional help from some proteasome-like structures and some additional enzymes, turns those into little peptide fragments, again, slightly different protein blender, and late in this pathway, just before this reaches the surface of the cell, these two types of vesicles actually fuse with each other. This is a pathway that's fairly unique to the antigen-presenting cells. Not all cells in your body are good at doing this way. So we're going to go over those in a little bit more detail. First of all, the proteasome. This is why I called it a little drum thing. This is roughly, it's roughly a cylindrical shape. It's got a hole in the middle. The proteins enter through one end. They make temporary interactions on the inside of this drum, and then they come out as little peptide fragments. There's an immune-specific one called the immunoproteasome that basically does something pretty much the same. What causes a protein to enter the proteasome? Because that's always an interesting question. There's a small protein called ubiquitin. It's ubiquitous, meaning it's everywhere. Often just abbreviated UB because people get tired of writing out ubiquitin, and it's a hard word to spell because it's got a Q in it. That'd be a good Scrabble word if you had two U's. This is when you're inside the cell and you add like 3 or more little ubiquitins onto a target protein in a chain, you target that protein to the proteasome for degradation. It's one of the mechanisms that your cell has for saying, this protein is old. Basically, ultimately, it happens to all of your proteins as they get older. It's one of the normal turnover mechanisms for your cell. But ubiquitin is used to tag proteins for regular proteasome degradation. The immunoproteasome has some slight differences. I'm not sure that I care if you know what those are. They're just genetically slight differences in some of the beta subunits. So instead of beta 1, you have the beta 1 immunoproteasome version and the beta 5 immunoproteasome version and the beta 2 immunoproteasome subunits. These actually chew up the proteins in a very slightly different way, but it makes them a little bit more accessible to MHC. So it tends to generate fragments that are the right size for MHC as opposed to just randomly sized fragments that come out of normal proteasomes. Remember, your fragments have to be like 9 to 13 in length. 9 for MHC1, 13 for MHC2. Yes.
So does the ubiquitin, does it phosphorylate the protein, or does it just?
No, it tags it like a phosphate group would, but instead of a phosphate group, it's a ubiquitin.
Oh,
Yep. Yes.
This tagging with ubiquitin, can it happen anywhere in the cell?
Yeah.
Cool.
It's mostly cytosolic. Tagging with ubiquitin and degradation in the proteasome is usually a cytosolic process.
You wouldn't see it in like the ER very often, for example.
You shouldn't really see it in the endoplasmic reticulum, and you definitely shouldn't really see it in the nucleus. However, ubiquitin itself is found in the nucleus, and it's actually used as part of the epigenetic modifications for histone tails.
Wow, that's cool.
isn't that like the ubiquitin process is also used for like determining signaling pathways too, right, in the cell?
It can also be used for terminating cell signaling in some cases, yes. The best known functions for ubiquitin, though, are really for proteasomal targeting. That's like pretty classically how we discovered it in the first place. That said, we'll probably find more functions for ubiquitin as we keep doing research with it. It seems to be sensitive to how many. So if you just put one ubiquitin on a target protein, that's not enough to get it to the proteasome for degradation. 2 is not enough. 3 is basically the threshold. Once you put 3 ubiquitins in a chain, so like just a little tail of 3 ubiquitins, and you attach that to your protein, that's what targets your protein for degradation. Once your proteasome blender has done its job and you've generated a bunch of fragments, you have to get those fragments into the rough endoplasmic reticulum so that they can match up with and bind and be mounted in your MHC binding groove. So there's a transporter protein specifically for that, and it's the TAP1, TAP2 protein complex. This is just an ATP-dependent transporter protein that's in the membrane of the rough endoplasmic reticulum, and its job is to actually take some of those peptide fragments, use ATP, and actually pump those into the lumen or the inside of the rough endoplasmic reticulum. It has a specific affinity for peptides that are 8 to 16 amino acids in length. Gee. That's right in our magic range. In fact, it's right in our magic range for either MHC1 or MHC2. Because it was like 9 to 13. 9 for MHC1, 13 for MHC2. And it favors peptides that have hydrophobic C-terminal ends. So this is important because these form the anchor residues at position number 9. For MHC1 in particular. So this protein is really kind of optimized to specifically pump little fragments of, say, 8 to 16 amino acids from the proteasome or the immunoproteasome into the lumen of the ER, and it's specifically going to work best for the ones that are the right size and that also have the hydrophobic C-terminus, carboxyl end. Basically, just one end of that protein needs to be, or peptide fragment needs to be hydrophobic or nonpolar. From there, now that we've gotten those peptides into the rough endoplasmic reticulum, we actually have to load them into the MHC binding groove. And there's actually chaperone proteins that assist with this. There's a whole bunch of them, including calnexin, ERP57, calreticulin, and tapacin. And I don't care that you actually remember most or any of those of them. I would guess, I think I saw tapacin the most in your textbook, but I'm not terribly worried about that. These chaperone proteins help your MHC fold properly. Again, tertiary structure is going to be really important here. For MHC class 1, which is the one we're mostly worried about right now, you don't have an alpha chain and a beta chain, you've just got the alpha chain, beta 2 microglobulin comes later. But these other proteins, the chaperone proteins, help to fold MHC class 1 and physically get it reasonably close to that TAP transporter that's actually going to be pumping those peptides in. So it puts them in the right place to kind of catch some of those peptide fragments as they enter the rough endoplasmic reticulum. From there, there's also an enzyme, the endoplasmic reticulum aminopeptidase, ERAP1 or ERAP1. It trims longer peptide fragments from the N-terminal end, not the C-terminal end. So it chops off the head ends of the proteins instead of the tail ends of the proteins to make them fit if they were a little too long. Because TAP is going to bring in fragments that are up to 16 amino acids in length, and that's a little bit too long for MHC class 1. So this enzyme helps trim them down to the final length. Now that you've got your peptide loaded into your MHC class 1, this MHC complex is just going to transition through the Golgi body and get transported to the surface of the cell, where it's going to be embedded in the cell membrane through normal pathway. Nothing at all unusual there. Yay. Send it to the surface and stick it up there. Good enough. Exogenous pathway for antigen presentation. This one, we will talk a little bit more about some cell biology so that we can make sure that we actually understand a little bit. So when your cell eats or drinks substances or imports substances from the outside world, the first stage of that process is what's called an endosome. The cell membrane is actually going to form what's called an invagination, so it's going to start to curve inward and create this depression. And eventually that depression is going to get deeper and deeper and deeper until it forms a literal bubble out of the cell membrane, kind of at the surface, and that'll eventually pinch off and form a little closed vesicle. That little vesicle is an endosome. Those endosomes usually fuse with lysosomes in cells, and lysosomes are famous for being the degrading organelles of your cells. They're the stomach of your cell. They're actually a pretty good analog for a stomach because they're acidic. The pH actually is going to continuously drop from about a 7.5 when it's outside the cell to down to a 4.5 in the latest stages of the endosome and lysosome fusion. It does not use hydrogen peroxide, it just uses hydrogen ions, H+, pure acid. So as it bathes the incoming proteins in literal acid, that's going to be enough to start breaking them down, ultimately until they get to the size of about 13 to 18 amino acids in length, which is precisely the right size for MHC class 2. Meanwhile, back at the rough endoplasmic reticulum, you had a ribosome that actually started your processing of making an MHC class 2 molecule. Initially, those are going to be associated with an invariant chain. So when we talked about the B cell receptor, when it was mounted on the surface of the cell, initially it was the 2 light chains that were invariant, and those were like a temporary placeholder. You're also going to have temporary placeholders when you're making MHC class 2. That will ultimately get taken care of and replaced with fully functional MHC class 2. But this invariant chain basically kind of sits in where the peptide should go in the normal binding site. And that's important because what it's doing while it's kind of just sitting there is helping the protein fold to its proper final conformation. Because for MHC class 2, you've got the alpha chain and the beta chain, and they have to come together and fold basically around this invariant chain to make the groove the right shape. And it's going to be a little bit wider and a little bit flatter and open on the ends compared to the MHC1 binding groove.
So it's just like a placeholder?
It is just a placeholder, yep. It's a placeholder for the antigen. So in your antigen presenting cells, which is where this process takes place, the Golgi vesicles that have this pre-made MHC class II with the invariant chain antigen placeholder, those are going to fuse with those late lysosomes. So you've got the lysosome coming in from the surface of the cell and digesting and degrading the peptide, and you've got that MHC2 coming from its normal pathway, a lot like MHC1. Those two vesicles are going to meet up and fuse with each other, and that's how your MHC class II is going to get its peptides. This whole process actually only takes about 1 to 3 hours. So if you're wondering, from the time you get a vaccine to the time when your antigen presenting cells start showing this or start having the ability to show this off, it's only 1 to 3 hours. That's surprisingly fast. It takes considerably longer for them to actually functionally turn on your T cells. That's good, because remember, your T cells kill you. Slowly, in little bits at a time, but that's what they do.
I remember my doctor telling me that like it'll take like 1 to 2 weeks for it to like fully take effect. Is that?
You reach, because your cells, after they're turned on and activated, they have to go through multiple rounds of mitosis. That usually takes 3 days up to and continues for a couple of weeks. So yes.
So at this point we're not like.
At this point, we haven't even turned on a T cell. This is just like, oh, look, now I have the ability to go show the T cell. This is what I found. Look, 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 this is what I found.
It's just.
Isn't it cool? Do any T cells recognize this? That's literally the job of an antigen presenting cell, is to capture antigen and go show it off to all the T cells. And literally basically say, look what I found. Can any of you T cells out there in the lymph node recognize this thing that I found? Because if so, we have a major problem. If none of you can recognize this thing that I found, maybe it's a normal thing. Okay, fair enough, never mind. I'll go away.
Yeah, but this process happens like right after we take the vaccine?
This process happens right after you get the vaccine. So yes, within a few hours, your dendritic cells have already discovered that there is a problem in that area and have started to acquire antigen, process it, present it, and run screaming back to your lymph nodes.
That's cool.
Meanwhile, while that's all going on, your innate immune system is also on the scene doing its thing, trying to send macrophages and neutrophils in to try to clean that up. If you think about it, that makes vaccine design a really interesting thing. You have to provide enough of the stuff that it overwhelms your innate immune response and gets your adaptive immune response involved. So no wonder your arm is sore. You have to get a pretty good dose of whatever it is. You're also injecting it kind of deep into muscle tissue, which is not normally where that virus would be. Like most of the viruses that infect us seem to be nasal or digestive.
Is that why they do some nasal sprays for vaccines?
That's why they do some nasal sprays for vaccines. Yep. What we know from experimental evidence is that if you actually mimic the normal route of infection, you get a more realistic immune response, and that seems to protect you better. At least in part because it seems to trigger more of the memory cells to actually live like in your nose where you need them, as opposed to in your upper arm where the virus is not going to infect you. Right now, my upper arm has been very well trained to recognize some COVID. That does me very little good. I mean, I know I've got lots and lots of memory cells. They're just not in the optimal place. So if we actually design our vaccines for nasal sprays, it does have an advantage. However, we've had some difficulty in making all of them as effective. So.
I feel like I only ever had this as a kid.
Yeah, they do that because kids are afraid of needles, and they don't care if adults are afraid of needles or not. They just expect us to suck it up and just deal with it.
Petition for adult nasal spray.
I would petition for adult nasal sprays. One of my first things that I did as a graduate student in terms of research was actually looking at, depending on where I gave my mouse a shot, if I did it like in the intravenously or if I just gave it to him in the peritoneum, like loose into the abdominal cavity, it made a difference into what types of cells got targeted. So it really does actually matter, and we don't really fully understand why. We know that it does. We're not clear on the mechanism, but that's somebody's job, is to untangle that and try to figure that out. That invariant chain, the antigen placeholder, its job is to prevent peptides from binding into the groove too early while the MHC class 2 is still in the endoplasmic reticulum. So remember, for MHC class 1, the chaperone proteins helped the MHC1 to fold and got it close to TAP. That's not what you want to have happen for MHC class 2. You want to keep MHC class 2 kind of away from TAP and folding in its own little cellular location, and this invariant chain helps keep it from binding to a peptide prematurely so that it can pick up an exogenous peptide later. Order matters. It uses sorting signals in the cytoplasmic end to help direct it to the endocytic compartments. So this is partly how you get fusion between the Golgi vesicles and the incoming endosomes. And that invariant chain, the II, it gets degraded in the endocytic vesicles too. So basically, you take this lysosome that's like a pH of 4.5, and you're going to merge that with your MHC class 2, which means your MHC class 2 is going to get chewed on by acid. It's designed for that, actually. But that invariant chain that it's carrying, the temporary antigen placeholder, that's going to get degraded. And it's going to get replaced by whatever antigen the cell happened to have eaten.
I never knew that II stands for invariant.
Yeah, in this case, II is invariant chain. It's also known as CD74. You do not need to know that it's CD74. I'll probably start pointing out the CDs because there's a whole ton of them, and we're going to start seeing where some of these come into play in the next chapter in particular. But for now, that's just one of the, I don't know, couple hundred CD molecules that we've designated. So the graphical version of this is more helpful to me because I have trouble like visually, mentally thinking of things. I don't think in pictures. I don't know about y'all, but some people do, some people don't. I don't. Therefore, I'm extra reliant on having good pictures to describe what's going on. So at the surface of your cell eats that antigen, whatever it may be, coming from outside the cell, whether that's bacterial or parasitic or fungal in particular, because viruses are usually intracellular. It gets engulfed in an endosome. That endosome fuses with some lysosomes, and the pH starts to go down. The more lysosomes that fuse, the lower the pH is actually going to go. This antigen is going to get chewed up into little peptide fragments. Meanwhile, your MHC class 1 is made back here in the rough endoplasmic reticulum, transported with its invariant chain, the II thing. It's going to get transported through the Golgi complex until it gets to about this point. And then it's going to merge with that lysosome, endosome thing, and the MHC class 2 containing vesicles are going to merge together all in the same place. The invariant chain gets digested, and you replace it with a little piece of the peptide. And then that complex gets sent to the surface of the cell. Good? The biggest difference between MHC class 1 processing and MHC class 2 processing is when the antigen gets loaded. Whether it's in the rough endoplasmic reticulum for MHC1 or whether it's in the late endosome stage for MHC class 2. Okay, so this is basically where we started. Up at the top, on the green side on the left, what you have is the endogenous antigen. That's some sort of cellular protein, whether it's the cell's own protein or whether it's a viral protein, doesn't matter. It gets degraded in the proteasome because it has been tagged by ubiquitin. Excellent, good, okay. Those little peptide fragments that come out of the proteasome get pumped into the endoplasmic reticulum by the TAP protein. There in the endoplasmic reticulum, you've got protein synthesis and folding going on, and your MHC class 1 and MHC class 2 molecules are both getting folded. MHC class 1 picks up its peptide because this happens over close to the TAP transporter. MHC class 2 takes place somewhere else. And it picks up its invariant chain to protect that binding groove. Both of them then get sent to the Golgi complex, where they start transporting toward the cell surface. But along the way, for MHC class 2, you actually get fusion with some of these incoming endosomes that fuse with lysosomes, so very acidic environments. This degrades that invariant chain, and then your invariant chain gets replaced with actual antigen that had come from outside the cell, and that ends up at the surface of the cell.
So the clip is only there to degrade the II.
The clip is actually a fragment of II that remains after the rest of it's been digested away.
It does. And what does the clip do?
Dissociates, it just dissolves out of the binding pocket and gets recycled. That's how it's supposed to happen. There are some exceptions. Of course, because it's immunology. And nothing about trying to fight off invaders is like simple and straightforward. Your body, since we're all mammals, we've all got this, it's got an amazingly complex system to try to defend us against all of these various things that would like to kill us. Actually, most of them don't really want to kill you. They just want to take over your body. Killing you is probably, from an evolutionary perspective, a bad idea. You just killed off your host. Great, now you can't spread to anybody else. That is why herpes is absolutely the pinnacle of pathogen evolution, in my opinion. The herpes viruses make you sick, but not that sick. You're not sick enough to stay home from work if you've got a herpes virus infection. You have a cold sore. It doesn't look very good, but you'll live. You don't even particularly feel sick. Most herpes virus infections don't even cause symptoms. They just hang out in your body and are completely and totally invisible to you, the human host, which means you go about your normal life, spreading them and sharing them with everybody you come in contact with. You infect all kinds of people with your herpes, and everybody in here has some form of herpes. Congratulations.
I heard that sometimes it stays in your body until maybe 50 years old or something, and then it shows up.
Herpes has an incredible ability to be latent in your body, so to remain hidden and not become active. Part of the thinking about that is that normal immune system patrolling of your body tends to deal with little spot recurrences and flares that do try to pop up, and it's just enough to quell them, and then as you get older, your immune system becomes less efficient at doing that. So they break through more often. Okay. So MHC class 1 is necessary to present antigen to CD8 positive T cells, right? Been drilling this into your head. Everybody's got that. Yes? Okay, good. So that means that viruses would actually have to infect antigen presenting cells. Oh, well, that's not so good. We don't want our antigen presenting cells to actually get functionally infected with virus, but the dendritic cell's job is to sample its environment and ingest things, and that may include some, well, viruses. But that means that that's exogenous antigen, and that should take the MHC class 2 pathway. So how do we get virus peptides from outside the cell onto the MHC1 on the internal pathway? Oh, well, that's a sticky problem.
So how do we get the virus inside of the cell without it?
How do we get the virus proteins inside the cell without functionally infecting the cell? Yes. That is definitely a problem. How do you get the proteins without getting an infection?
Can't you just break it down extracellularly?
Can you break it down extracellularly? No, not without degrading your own extracellular matrix.
You couldn't just like fire off. Like,
You don't want to just dump some acid loose into like under your skin, do you, randomly? Probably not a good
Just
option.
A little bit.
Just a little bit.
Is it true?
No. So there's a couple of options. One of these is to actually just take some of that TAP protein and actually stick it in some endosomes and have it pump peptides out of the endosomes and loose into the cytoplasm. So those MHC class 2 proteins, those peptides, would then be in the cytoplasm. If the TAP pushes them out into the cytoplasm, then the other TAP on the endoplasmic reticulum can then pump them in to get them on MHC1. That works. That is a simple solution to a very complex, thorny problem. I like that solution. That one's my favorite. I like that mechanism because it's easy and it makes sense. The other is to actually take some of that MHC1 and transport it to the endosomes. That works too. Why is it just the MHC2 vesicles that fuse with the endosomes? Couldn't MHC1 vesicles also fuse with the endosomes? Answer, yes, they seem to, sometimes. Not as often. And they're not protected with an invariant chain. They've already got peptide, so they would have to reject their existing peptide and acquire a new peptide, or somehow never have gotten a peptide in the first place. This is a little bit tougher, but it also seems to happen. Both of these are two of the main options and the ways that we think this actually plays out.
So you will have the virus, it will
So
fuse with the cell, go to the
endosome,
endosomes,
get degraded,
and then set.
and then hopefully some of those peptides will end up on MHC class 1.
Okay.
Yep. And that's where we will pick up on Thursday. We will also start chapter 8 on Thursday because I've only got 3 slides left in this chapter. We made good progress today.
Thank you, Dr. Steele.